You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Dr. Andreas Werner. He's part of the Institute for Cell and Molecular Biosciences at Newcastle University over in the UK. So, Andreas, thanks for coming. How are you doing?
I'm very well. Thank you, Richard. Thanks for having me and giving me the opportunity to talk about my research, my science.
Yeah. Well, I see it's related to the genome. So, can you give a brief description of what research you're working on?
Oh, yeah. So, very generally, I work on two different topics, and they are not really related. The first one is rare diseases that affect the kidneys, and the second one is genomics, and more specifically, noncoding RNAs and evolution. And in the context of this conversation, I would suggest that we focus on the second one, on the genomics and the non-coding RNAs, if that's all right with you.
Yeah, what are non-coding RNAs as opposed to DNA for the layperson?
Right. So let me explain that a little bit in more detail. The extent of non-coding RNA is going back to the Human Genome Project, which basically enabled the technology and our that brought about our understanding of these non-coding RNAs. And I think if we talk to a layperson, it's pretty much understandable that molecular biology and would basically consist of genes, and then these genes would be transcribed into RNA, which are kind of blueprints for proteins, and the proteins do the job. And that was pretty much the perception of how a cell would work, probably up to about the 90s and of last centuries, pretty much a mechanistic perception of a cell. And at that time, the Human Genome Project actually was started. And I remember that at that time I thought, well, this is not really worth the money, because there are genes, and we know that there are loads of areas that do not encode for a gene or a protein. And I thought it wasn't worth the money. And we could already at that time sequence genes, and we could simply forget about these non-coding areas, this junk DNA or gene deserts. Gladly, the committee, they didn't listen to my concerns and went ahead anyhow. and there are two major outcomes that I would like to focus on from the Human Genome Project. And the two features are that there are loads of repetitive sequences, that we have millions of copies of the same DNA sequence, and these resemble old viruses or old kind of elements that can invade the DNA, and for one reason or the other, we're able to kind of invade our genome. And gladly, most of these viruses, they became in ineffective and inefficient and non-functional, but remain in the genome kind of as ruins. But now we come to your question about the non-coding RNAs, that much of these non-coding areas are actually transcribed, and many of those give rise to these non-coding RNAs. And that's an additional outcome of a spin-off, basically, of the Human Genome Project, that the sequencing technology got so advanced that we are now able to sequence an entire transcriptome within weeks for maybe $1,000 or £1,000. So this is really becoming affordable.
What do we think the role of the non-coding RNAs are? If they're not coding for anything, what do they do?
Well, that's a big debate. Initially, when we realized that there was so much RNA, there were voices that they are regulators of gene expression. They are important for many, many things, for tuning the expression, for degrading RNA transcripts, etc. So they were thought that many of these RNAs have a really important function. And fair enough, if you knock some out within a cell, you may end up generating a phenotype and affecting the fitness of the cell. Kind of having thought of these huge numbers of non-coding RNAs and the difficulty of establishing a function for many of those, I kind of tend to take a slightly different approach. So if we look at what these viruses and repetitive sequences provide to the genome, it provides vast amounts of information, these truncated proteins, but also regulatory sequences that actually start transcription, drive transcription. And if we look at all these areas of non-coding DNA that are transcribed, we can look at these transcripts as kind of emerging genes, emerging proteins, not necessarily proteins, but just basically transcripts that are released into the cell nucleus and into an environment. And you can look at that like kind of babies that are generated and are basically kind of tested, and they are released into the cell, and most of them they are not stable enough to survive, or they lack certain features that they are not processed like a normal RNA.
Where do these non-coding RNAs reside? They're not residing on the DNA itself. They're attempting to transcribe parts of the DNA that normally aren't transcribed. Where are they? Where do they sit in the cell? Where are they formed?
Well, they are transcribed at a low level, and transcription as such is not 100% tight, that it only transcribes genes and then stops, but there might be kind of ripples of transcription around genes. There are areas that maybe just kind of a buildup of transcription machinery that produces a very quick kind of run through a non-coding area, but then falls off again. So kind of spurs transcription that is more kind of serendipitous and random than driven by a specific promoter. But the important thing is that many of these non-coding areas, they contain DNA sequences that drive transcription, that are old promoters that were used by viruses originally, but the whole rest of the virus is kind of non-functional, but the promoter still kind of flickers here and then and provides transcripts. And if the resulting transcript kind of, for one reason or the other, maybe it clinches to another transcript, it clinches to a protein, kind of makes itself indispensable and starts kind of regulating something or inhibiting or enhancing something, then that transcript may acquire a function. And you probably can see how difficult that is if some kind of naive RNA enters the cell in the nucleus and then has to basically survive and define itself as useful to the cell, as a regulator or even a structural component. that's really difficult. So most of these transcripts will be non-coding, short-lived, being degraded without much of a biological function. But it is necessary to the cells, to our organisms, to provide this possibility that we can innovate, that we can generate new genes and new transcripts.
What kind of cells are you looking at? Are you looking at human cells? And if so, what kind? And, how are you figuring out that these things even exist or what their function is?
Right. We are looking at human cells, also at mouse cells, but that doesn't really matter. I would consider both mice and human as very complex organisms that, interestingly, share much of the structural and the functional components. The proteins are very, very similar between mouse and human. What is mostly different are exactly these non-coding areas. So that brings me to another point. These non-coding RNAs, they are very, very dependent on the context. Often you find non-coding RNAs that do something, and research often focuses on diseases. So you look at a non-coding RNA in a liver cancer, for example, you look at the expression level of this non-coding RNA, and a high level of this RNA is very bad for the outcome. The patients with high levels of this non-coding RNA have a low survival chance. Now you look at the same RNA, the same non-coding RNA, and look at it in a kidney cancer, it's exactly the opposite. Those with a high level of this RNA, they live for a long time, and those with a low level, they die very quickly. So the functioning of these non-coding RNAs is exquisitely context dependent, and therefore it's often difficult to compare a function of a non-coding RNA in a mouse and compare that to the functioning in a human. So this probably indicates that our differences to animals, etc., are mostly down to non-coding RNAs and not to the protein coding genes.
Why don't you get some human cells and work on them instead of mouse cells? It's going to be it's not inside the organism. I mean, it's in a dish, right? Or in a microenvironment you create in the lab. So why not get human cells, stem cells, skin cells, something that it's really not invasive to get?
I think we're not quite there yet. So the problem is that whenever you look at a specific RNA or a specific kind of gene cluster, a regulatory network, you can focus on one or two or three or five components, and there are hundreds of others. So if you kind of try to recapitulate something from a human cell and a mouse cell, you're probably on a lost case there, because you simply are unable to control all the different factors that would guarantee what you see with your addition of the non-coding RNA.
Okay. Well, so is your model a mouse model, or what kind of model is it? I don't understand.
So let me kind of become a bit more specific about these non-coding RNAs. If you look at many of these non-coding RNAs, they are particularly highly expressed in testes, which indicates that there might be something going on with respect to changes to evolution, because where evolution happens in testes, and in testes you see most of these non-coding RNAs. Difficulties with testes, with sperm cells, you can't basically culture them. So everything that we try to kind of recapitulate with non-coding RNAs might be in a wrong context, and we may produce wrong conclusions. So I would suggest that if you are interested in the functioning of a specific non-coding RNA, you investigate that in the specific cancer or in the specific organ that you want to understand its function, and you get as close as possible in establishing a model system for that, a cancer cell line that reflects that, let's say a liver cancer, and you stick with that. And then you try to extrapolate that to human liver and nowhere else. So it's really a tricky call there.
Is the name non-coding RNA a misnomer? I mean, what function do they have? Have you been able to elucidate anything about them?
We know about a number of functions. They can basically provide a scaffold for enzymes. So because they are so compliant, they can actually form a very versatile scaffold for proteins. And then in addition to that, because they are nucleic acids, they can anchor to RNA, to nascent RNA. So by interacting with a nascent transcript, they can deliver a protein component to that active locus, and that protein component can either facilitate or inhibit the transcription, and hence that non-coding RNA has an influence on gene transcription of that particular target gene. Alternatively, there is a particular family of non-coding RNAs which are transcribed in the opposite direction of protein coding genes. And if you want, I can expand on that a little bit, because that's a very important area of my research, the so-called antisense transcripts.
So these particular RNA fragments or microRNAs, they're not involved in transcription. They appear to be involved in assisting other elements to be transcribed or expressed, or enzymes to participate in reactions. Is that they're kind of helpers instead of active coding elements or transcription elements?
Yes.
Is that what you're saying?
That's exactly correct. And that kind of feeds into my kind of analogy with kids with developing kind of RNAs that have to make themselves useful. So they will easily make an impact if they serve a regulator, they up or down regulate an essential gene. If they manage to do that, they become very easily indispensable for a cell if it up or down regulates an essential component of that.
All right, so go ahead into the other area of research that you're talking about. Continue to describe it a bit.
Right. So I was thinking of looking at these antisense transcripts that are transcribed in the opposite direction of genes. And this is really interesting because, again, these transcripts, they're expressed in testes, and potentially they could hybridize and form double-stranded RNA with the sense transcript. And this is a structure that also resembles a virus. So you have this kind of double-edged sword that you have these two transcripts that can have a regulatory function, but also it can be interpreted by the cell as a virus. And this may have huge implications in diseases, that these non-coding RNAs can actually trigger disease, and by mimicking a viral infection and generating an underlying inflammatory phenotype. So that's another aspect of these non-coding RNAs.
So how close do they look to exogenous viruses?
So if you infect a plant cell or a C. elegans cell with double-stranded RNA, you will elicit an immune response that is for these species and organisms an enzyme that chops the virus up into small RNA bits. In human cells, that works differently. If you infect the human cell with a virus, proteins that recognize double-stranded RNA viral structures will generate an immune response. And you ask whether they resemble each other, and the answer is yes. So you can trigger these responses that are usually directed against the virus by releasing the brake on the transcription of these gene deserts, of these repetitive elements that eventually form transcripts that hybridize to each other, form double-stranded RNA, and usually they are kept within the nucleus. But if that kind of protective barrier is overpowered, it will leak into the cytosol, and then it gets recognized and it triggers the same response as a virus.
Well, what does a virus look like? I mean, outside the cell, I know it has a protein coat, but once it injects its RNA or DNA into the cell, then does it closely resemble these double-stranded RNAs?
Yes. Basically, every virus in at certain stages during its life cycle, undergoes a phase where there is double-stranded RNA. Now, the cell has obviously various possibilities of attacking the virus, but the structure of double-stranded RNA is a structure that the cell recognizes as foreign and attacks it. And the way it attacks it, there are a number of proteins that very specifically can distinguish between DNA and RNA, because if you recall the double helix of DNA is slightly different as compared to that of RNA. And it takes specific protein that can kind of recognize the size of the groove of that helix. And by distinguishing between the size of the grooves, they can distinguish between double-stranded RNA and DNA. And it takes a certain length of double-stranded RNA. So if you have short pieces of double-stranded RNA, let's say 20, 30, 40 base pairs, that probably doesn't trigger this response. But if you go longer, which is the size of viruses, long double-stranded RNA greater than maybe 40, 50, and then hundreds and thousands of base pairs, that will be recognized by these proteins, and they bind to these structures, and then that immune response, an interferon-mediated immune response, is triggered, which eventually leads to apoptosis.
All right. So what is the goal of your research? Just to understand the function of the microRNAs, to create them and see how they influence cells? I mean, what is it?
Right. The goal of my research is basically kind of informed by this kind of bias between intrinsic structures that are able to form double-stranded RNA and a viral response. So the fact that the cell intrinsically generates molecules that can kill the cell is kind of beggar's belief. So it's absolutely counterintuitive. Nevertheless, the cell does that, and we want to understand that better, because the hypothesis is that under normal circumstances, these double-stranded RNA molecules are maintained in specific compartments in the mitochondria and in the cell nucleus. But we hypothesize that under certain stress conditions, and that stress can be very kind of low, but recurrent. So also, for example, if a cell ages, that we see an increase in double-stranded RNA that leaks out of these confined areas, cell nucleus and mitochondria, into the cytosol, where it triggers weak initially, but then a stronger immune response, and generates a phenotype that makes the cell much more susceptible to disease. And there are preliminary results that predict that, for example, in cancer, but also in autoimmune disease or neurodegenerative diseases, you have an underlying phenotype, inflammatory phenotype, that we hypothesize is brought about by double-stranded RNA that leaks out of the usually well-contained compartment. And we want to prove that. We want to find very specific methods to measure this double-stranded RNA, to measure that double-stranded RNA in patients that either have specific cancer or are affected by neurodegenerative diseases.
How similar morphologically is double-stranded RNA versus DNA? Sounds like a bad name for it. I don't know. How do you know that there's even a difference?
If you look at the DNA double helix, you have a large groove and a small groove. So the helix basically has two grooves where a protein can access and basically feel the base pair composition within that groove. And this works well because the DNA has one relatively wide groove where a protein can access that information. This is not the case in RNA. So the RNA is basically more hidden. The base pair composition is more hidden in double-stranded RNA than it is in DNA, and hence protein cannot kind of feel the the base pair composition, the base pair sequence of a specific double-stranded RNA sequence. So double-stranded RNA is much more of a molecule that has kind of biochemical properties and less of a sequence with A, T, C's, and G's as the DNA.
You can tell the difference experimentally pretty easily between double-stranded RNA and DNA, or is it very subtle?
There are antibodies that recognize double-stranded RNA specifically, and they don't interact with DNA at all. So it's very easy to separate double-stranded RNA and DNA. Yes.
And do you understand the mechanism on how double-stranded RNA or even non-coding RNA is generated? And I believe you said it was generated in the mitochondria, or where is it generated?
Well, there are two major compartments in the cell where it is generated, and you named the first one, which is the mitochondria, and that has to do that the mitochondria have a very short genome that is transcribed from both directions, and despite the strands not being have the same stability, there is lots of double-stranded RNA being formed. The mitochondria, they have a very efficient protective mechanism to keep the double-stranded RNA within the mitochondria, so to avoid the double-stranded RNA leaking out into the cytosol. The same applies to the second compartment where double-stranded RNA is being produced, and that's the nucleus. And there are experiments when you kind of release the brake from transcription and enhance with drugs, spurs transcription, you tend to overpower the system, and the protective system to contain the double-stranded RNA, and there is an increased likelihood that double-stranded RNA leaks into the cytosol, where then it gets recognized and triggers a interferon gamma response. So you can generate experimental situations where you overpower the intrinsic protective mechanisms and systems that maintain the double-stranded RNA compartmentalized, and then you can measure that this immune response, this innate immune response, is being triggered.
What would be your ultimate and thrilling goal to achieve in the next couple of years with the research? What's really first on the list for you to figure out, and what will be the implications if you did?
Right. That's a very ambitious question, I think. You see, I'm interested in these antisense transcripts that I briefly touched upon in the middle of our conversation. And just because these antisense transcripts, they can and do form double-stranded RNA, I'm really puzzled why nature actually invents a regulatory mechanism or a system where it uses these antisense transcripts that is so dangerous for the cell itself.
Do we understand apoptosis? Because maybe we don't understand it fully, and this is a necessary component of it.
No, I don't think it's kind of induced apoptosis. I think it has to do with evolution, that it provides a mechanism, a possibility of comparing expression levels of certain transcripts. And eventually it allows a distinction between different types of RNA and eventually allows the detection of mutations or of poorly transcribing, poorly performing transcripts. And I think that's very, very important in the context of evolution to develop such a system, that a complex organism like a mammal can screen its genome for faulty genes. And I think these antisense transcripts provide a mechanism that allow them to do that at a very basic level. But that's my kind of hypothesis, and I think the ultimate goal of my research would really be to actually nail that down and confirm my hypotheses.
Well, it sounds like it could be a regulatory type thing. You know, like you said, it's evaluating the expression of multiple elements, and depending upon the conditions in the cell, it forms and maybe intervenes. You know, it doesn't just catalog or maybe through the immune response, that's how it's attempting to modulate the expression of various things going on in the cell, and it's an alarm for things going wrong or going in the wrong direction. I don't know.
True. I think you have to take into account that the organ specificity, not only so in testes where you have most of these double-stranded RNAs, they may have a completely different role as compared to in a cell, in a somatic cell. And I agree that there could be something like an alarm system in a somatic cell, but in spermatocytes, that's certainly not the case, right?
So, yeah, I guess it's a big puzzle. So I understand why it's intriguing you. So what's the best way for listeners to learn more from you, and then also from ancillary sources, so they can get a handle on what this is?
So I'm very happy to be contacted in any way. You can either call a phone or send me an email, visit my web page, read my papers. That's absolutely fine. My email address, I can give it here. That's my name, Andreas, and then a dot, and Werner at NCL for Newcastle .ac.uk. And I'd be more than pleased if I get feedback and people want to learn more about the topic.
Are you a lone wolf on this? I mean, how many different scientists appear to be working on similar type stuff around the world? Are you able to collaborate with a lot of people, or is this so niche that you're one of the rare ones that works on it?
Absolutely. I collaborate with a number of people here in the UK. I collaborate with people in Sydney, in Finland, and this is really an emerging field. Interestingly, the occurrence of double-stranded RNA has only emerged maybe a year ago or 2 years ago. So this is really a kind of a new line of thinking, really, that these molecules can actually have a cellular role and are key players in many processes in evolution, but also in disease. So I think I'm convinced it's an emerging and a very interesting field, and I'd be more than happy to collaborate and discuss at various levels, whether that's kind of exchange of ideas, whether that's exchange of protocols, or absolutely anything is possible.
Well, very good. Well, Andreas, I appreciate you coming on the podcast.
It is a real pleasure to talk to you, and I hope to get some interesting input and ideas, and I hope we make some more progress.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.